You have fallen into Event Horizon with John Michael Godier. In today's episode, John is joined by Stephen Benner. Stephen Benner heads the Foundation for Applied Molecular Evolution, which he founded after serving on faculty at Harvard, ETH Zurich, and the University of Florida. His research combines two traditions in science, one from natural history and the other from physical sciences. His laboratory has been a leader in the field of synthetic biology, where it has redesigned DNA to better understand how these molecules work, generate new classes of diagnostics tools, and to open new avenues to disease therapy. Separately, he also created the field of paleogenetics. His work also guides NASA missions to search for alien life and how life originated. His most recent book is titled Life, the Universe, and the Scientific Method.
Dr. Stephen Benner, welcome back to the program.
My pleasure.
Now, Doctor, the contentious issue of Viking in 1976 and thereafter, where we landed two rovers identical, both equipped with life-finding missions, instrument packages designed to determine if there was, in fact, microbial life on the surface of Mars. There were three experiments in each package, and they were identical. And they all returned positives, basically. Give us an overview of what Viking found with those experiments and how it was called. We'll get it later into how that was called into question, which begs the question, why did we even send them if we weren't going to listen to the results? But
well, sure. Well, I'd love to put your audience into the frame of mind to ask them how they would go looking for life on Mars. Remember, there is an atmosphere. It's carbon dioxide. That's a source of carbon. Actually, the amount of carbon dioxide in the atmosphere is not so much different from what the amount of carbon dioxide is in the Earth's atmosphere. It's just that carbon dioxide in the Earth's atmosphere is a very small fraction of the total, but their carbon dioxide is pretty much all of the atmosphere of Mars, so the total pressure is pretty low. But, if you think that life requires carbon, and that's a statement that would hold true for lots of people, even today, then the life, if you think it exists on the near surface, has got to get carbon from somewhere, and it's easy to get it from the atmosphere. And if you look at plants on Earth, that's where the plants get their carbon. They do photosynthesis by extracting carbon dioxide from the atmosphere. They use light as the energy. They use water as a source of hydrogen atoms, and they spit out oxygen as the byproduct. And so that's why your planet is green and has oxygen in the atmosphere, because the plants are moving carbon dioxide out of the air into plant material, which is organic carbon. So the first experiment that they did was look for that very same process on Mars. They call it the carbon assimilation experiment. It's sometimes also called a pyrolytic release experiment. It takes a little bit of explaining why these experiments have two names, but the carbon assimilation is basically what they did. They put radioactive carbon dioxide over the surface of Mars and turned on the light. So carbon, has a couple of isotopes. Carbon-14 is radioactive. Carbon-12 is what we have. Not radioactive is the overwhelming majority of it. Carbon-14 decays 50% of the time. Its half-life every 5,000 years or so. And when it does that, it spits out a high-energy electron, which they can count. And so they put the carbon dioxide on the Martian soil, turned on the light, and they looked for the absorption of carbon dioxide into the soil. And they had to now later recover it to determine that the soil had absorbed carbon dioxide. We'll go into the details a little bit later, but what they saw was the absorption of carbon dioxide, about enough carbon to account for maybe 1,000 Earth-like cells per gram of soil. And, that's about the amount of life that you'd expect to see in a very resource scarce environment on Earth. If you go to Antarctica and you scoop up soil in some of the dry valleys, you have about 1,000 cells per gram. If you go to the desert, the high desert in Chile, the Atacama Desert, they're about 1,000 cells per gram. And so they saw pretty much what you'd expect to see. That was the carbon assimilation experiment. There was also something called the label release experiment. This is the one that was run by Gil Levin and Patricia Strott. And, it's sort of the opposite. So what they, instead of looking for the formation of organic material through the fixation of carbon dioxide, like a photosynthetic plant, they went and looked for the reverse. They fed food, radioactive food as well, to the surface and looked for the exhalation, the release of carbon dioxide. So they put radioactive amino acids on the surface of Mars. If they had carbon-14 isotope, making those radioactive as well, and they looked for the release of carbon-14 carbon dioxide, which is what happens if I put radioactive carbon dioxide on you. You inhale it, you use the oxygen from the air to oxidize it, and you exhale carbon dioxide. And they saw that also. Again, about enough to be accounting for, say, 1,000 cells per gram, if you assume that the cells on Mars do metabolism at roughly the same rate and of the same kind as you do on Earth. So the second experiment was life positive as well. They called this respiration. We call that respiration when you do it. The third experiment was actually quite agnostic for metabolism. They didn't really have much of an idea. They just said that, well, if the soil has a living microbiome, it will exchange gases with the atmosphere. They looked for a bunch of gases. Carbon dioxide was one, water was one, oxygen was one, methane was one. And so they humidified the soil, and they saw a relatively large amount of oxygen being released from the soil. They saw some exchange of carbon dioxide. That was a life positive result as well, but it also, the oxygen that was in the soil was also the oxygen that they needed to do the respiration of the food that was labeled. So three life detection experiments, one looking for photosynthesis, the movement of carbon dioxide out of the air into organic stuff, and the second looking at respiration, the moving of organic stuff back out into carbon dioxide, and then the exchange of gases, and they saw it. So there you are. So the question for your audience is, well, hey, if you were running these experiments, what would you do, right? Well, people say, you would go out onto cameras and declare you to discover life on Mars and book your airplane to Sweden to get a Nobel Prize. Well, that's not what scientists do. What we do, we're afraid to go out and say in public that we have found life on Mars because this is big. This is a big discovery if that's the case. So what you would do, and of course what they did, was they tried to perturb the experiment to get, a life negative result. So they sampled soil from underneath a rock, or they heated the soil to try to get rid of the activity under the theory that if it's a living system, if you heat it to more than the boiling point of water, you'll kill it. They tried stepped releases or timed releases of things. And all those results came back also consistent with the biosphere. So in the case of where they were looking for respiration, when they were looking for the metabolism of food, they heated the sample to 50 degrees centigrade. That's maybe 150 Fahrenheit. And the activity went down by a half. And then they heated it above the boiling point of water. It went down completely. The same result was seen with the photosynthesis experiment. So an experiment, as they tweaked them, they got life positive results again. And of course, these are hard to do, right? You don't go out to the grocery store or down to the Home Depot to pick up something you forgot to bring with you because you just decided you could use some other reagent or some other tool to look for life. You only have what you brought with you. And furthermore, this spacecraft, is 10, 15, 20 light minutes away from Earth. So you send a signal out, it gets there maybe 10 or 15 minutes later, and then it responds, and sometimes the equipment leaks. And this is rocket science. So the result is that over a period of a couple of weeks, they tried to perturb the experiments to try to get a result that would contradict life positive, and they couldn't find one.
Yeah, it was very stubborn. It was a very stubborn group of experiments. Now, they also had a gas chromatograph mass spectrometer that was looking for organics. And it also was stubbornly saying there is no organic chemistry here, apart anyway from the carbon dioxide. There isn't anything there. We later found that that was erroneous, and we have since found organics on the surface of Mars with the rovers, etc.
Mm.
And so that was wrong, which is weird because that instrument probably should have detected organics. Do you think it actually did, and they talked themselves out of it?
Well, that's right. So we know that organic molecules fall onto the surface of Mars all the time in the form of carbon-containing meteorites. That happens to Earth. We can collect them. You can buy them on eBay. Some of them are actually authentic. And so we know that organic molecules fall to the surface of Mars also. So the gas chromatography mass spectrometry, that's the instrument you're referring to. What they did was they went looking for those meteoritic organics in the hope that if they could understand what they were, they would understand something about the history of organic molecules on Mars. They expected to find them. However, the instrument was not sensitive enough to detect fewer than, say, 1,000,000 cells worth of organics. So if you're saying that the light detection results are detecting 1,000 cells per sample of organics, you wouldn't expect the gas chromatography mass spectrometry experiment to be sensitive enough to see those. In fact, you wouldn't expect the GCMS, as we come to call it, to be sensitive enough to see organics in Antarctica or in the high desert of Chile. So, but never mind, they scooped up the material, they put it into a little saucer, they heated that saucer up to 500 degrees centigrade. So the idea here is you have organics, they're sitting in the soil, they're not volatile, you can't get them out of the soil. But if you heat the soil to 500 degrees centigrade, you fragment them. It's sort of like heating popcorn to 500 degrees centigrade. You will smell it, and then you smell it because the organics in the popcorn are breaking down at these high temperatures. You waft the smell of the popcorn into a gas chromatography machine, and that passes the gases down a long tube and then separates them. And then you dump the material that comes out of the tube into a mass spectrometer, which is supposed to tell you the molecular weights of the fragments that are coming off of your burnt popcorn, or in this particular case, burnt life cells in the soil of Mars, or more likely, the burnt organics that are formed, come to Mars all the time by meteorite. Now, the joke of it is that they said, well, the story's a bit complicated for your audience. So the first thing that they found was they found freons, which are the things that you use for refrigerants. Now, they had brought those with them from the Earth, and they knew that because on the way out to Mars, they had turned the mass spec on and they had detected freon. So they knew that was a terrestrial contaminant. But what they also saw was a compound called methyl chloride. It's one carbon atom, 3 hydrogen atoms, and 1 chlorine atom. And that's a gas. It boils at minus 24 degrees centigrade. They saw it. They report it. You go back into the papers, you can see a nice mass spectrum of methyl chloride, CH3Cl. And they write in the paper, they say, well, we did not see this in flight out to Mars. So it's possible that methyl chloride could be indigenous to Mars. Those are their exact words. Now, it's a bit of a problem. The question is, why is there methyl chloride in the surface of Mars when it's a gas that boils at minus 24 degrees centigrade? Now, the short answer to that question we know now is that the soil also contains an oxygen called perchlorate. It's chlorine atom bonded to 4 oxygen atoms. It doesn't do anything at low temperature. You can put it on your desk and it doesn't do anything at all. But if you heat it to 500 degrees centigrade, it sets organic materials on fire. And one of the minor products of that is methyl chloride, CH3Cl. So they observed CH3Cl. That actually means that they observed perchlorate burning the organics. And we now know, as you just mentioned, Mars has large amounts of organic molecules in the soil. So they burned them. They set them on fire, basically. But since they didn't know, well, it's an interesting story. They were aware that it was possible that the soil contained nitrates. And if you ever have made gunpowder in your life, have you made gunpowder, JMG?
Not recently.
Okay, well, it's usually an adolescent thing, but the idea was that you put nitrate on organics, you also set them on fire. And they knew about that, and they had worried about that, but they hadn't thought about perchlorate. They were not to discover perchlorate on Mars until 2009. And so they couldn't figure out, and it didn't occur to them that the perchlorate might put chlorine into the organics to make a little bit of methyl chloride. But we now know that that's what they saw. But at the time, they didn't know that, so they thought that they hadn't seen any organics except this very annoying methyl chloride, which they didn't know where it came from. So what you can do is it's fascinating to watch the papers as they come out in succession. The first paper that shows methyl chloride is written by the lead, a man named Klaus Biemann, and a very nice Austrian chemist who worked at MIT, and he said that methyl chloride could be indigenous to Mars. The next paper that came out, he said it was a terrestrial contaminant. And the next paper that came out that reviewed this result said that the methyl chloride was a cleaning solvent. So this was written by a man named Norm Horowitz, a biologist at Caltech, and we chemists like to make fun of biologists because they do not know any chemistry. So Norm Horowitz has confused methyl chloride with perchloroethylene, which, by the way, is a cleaning solvent. If you go take your suit to the dry cleaners, that's what they use with it. But by the time that paper comes out, nobody makes the connection back to the original paper. They say, in fact, you'll see this in review articles, you'll see this in textbooks, that the Mars Viking GC mass spec detected only cleaning solvents. What it did detect instead was methyl chloride, which, as we now know, is the combustion product of organics and perchlorate. Now, imagine yourself right back in 1976. You don't see the organics that you expect to see coming from meteorites, and you see oxygen coming off of the soil. And so what you say is, there must be a strong, powerful oxidant in the soil that toasts the organics, any organics. In fact, the story evolves over time. It becomes that the soil contains a powerful reagent that generates oxygen and that burns organics. In fact, the soil is so oxidizing, it is self-sterilizing, which is what the community said. And so by the time I did not get involved in this until 1999 because I was too young to be involved with it in real time. But the bottom line is that by 1999, everybody knows that the Martian soil is self-sterilizing. If you put life there, it would just burn because of a mystery oxidant, which Viking had discovered, the same mystery oxidant that had burned all the meteoric organics. So what you see is a misinterpretation of a GC mass spec result, which then propagates into the literature, but for good reason, right? If you're in 1976, if you're about to go out and announce that you discovered life on Mars, right? And you'd be really embarrassed if a week later some high school kid points out that you made a mistake, right? The first thing you do is grab at any negative result that you can think of. And so when the result came back that the organics, the soil of Mars contained no organics, Jerry Soffin, who ran the mission, said right off the bat, that's the ball game. No organics, no life. And now you can go out before the camera and say, hey, I'm sorry. We'd hoped there was life. There is no life. There's no organics. And then you get to go home for dinner.
Why spend the money in the first place?
Well, yeah. So the obvious question is, if you're going to go to Mars and you're going to discover life, if you're afraid to say so, why do you buy to go? And by the way, there's a very nice lady, Inga ten Kate, who's Dutch, and she just wrote a paper which came out a couple of days ago, where she pointed out that the bias in science is to worry more about false positives. That is, saying that you have found life when you have not. It's more likely that you worry about those than we worry about false negatives, which says that you didn't find life when you have. And that is, of course, because of how science and the community structures reward and punishment. Right? I mean, if you go out and say that you have found life, you get what happens to Gil Levin. I mean, Gil Levin was, and well, Gil went through this has a story. He says he's a good friend of mine, and I every now and then we take his slides and give his talk for him. If he was getting old, of course, by the time I knew him, but Gil initially found life. And in fact, I can produce for you a piece of paper showing the initial trace of the release of carbon dioxide from the surface of Mars with his signature on it, as well as everybody else on the team. They had basically thought they had discovered life. But then no organics came through, no organics, no life. And so Gil didn't quite know what to do, right? He couldn't argue that organics were not necessary for life. He was not enough of a chemist to know that methyl chloride would be the product of perchlorate burning the organics that were actually there. He did not know of the perchlorate. And by the way, and nobody did a back of the envelope calculation to say that you wouldn't expect organics at this level from a microbiome that was living in a sparse research environment. And there was a guy named Robert Jastrow who was head of NASA Goddard Space Center. Only he persisted in this. And I have a great book of his. It was written in 1977 called "Until the Sun Dies." He has a whole chapter on this Viking result, and he says again and again in his career that the GC mass spec is not sensitive enough to detect fewer than 1 million cells, and the life detection experiments are sensitive enough to detect 10 cells or 100 cells. They're detecting 1,000 cells, and that's about what you'd expect. And he said that again and again, but at some point, the community moves on. So Gil was, of course, a little despondent by all this. But in 2000, when I was first came back from Europe, I was living in Europe. I first became aware of the results. I mean, I was dragged down to the Lunar and Planetary Institute in Houston, Texas, where the library has several shelves of the Viking mission. And so I'm a chemist, so I was paging through this in the evening, and it was clear. Two things were clear. First, they had detected life. Second, they were afraid to say so. And third, the GC mass spec was not correctly interpreted. And so I wrote a paper in 2000, there's a quarter century ago, saying that the GC mass spec data did not make the argument that they were assuming, everybody was assuming they made. Now, when Gil heard that, he returned to the fray. When perchlorate was discovered on Mars in the year 2009, he was emboldened. Then a guy named Rafael Navarro Gonzalez from Mexico City, he in 2010 published an incredibly detailed and entirely correct analysis of everything that went on Viking. Since he knew about perchlorate, he knew about the oxidation of organics with perchlorate, he knew that methyl chloride was one of their products. And then Gil in 2016 went back and said, okay, he wrote a very famous paper basically revisiting the life detection experiments. But what you see by then is that the culture has moved on. So, the textbook in 2005 from a good friend of mine, Jonathan Lunine, I have a copy of it around here somewhere, says that they didn't find any organics on Mars. They didn't see any respiration. They didn't see any photosynthesis, even though they did. But that's the story. NASA contracts a bunch of students to write what's called an astrobiology primer, which is supposed to give you general, knowledge about what's important in astrobiology. They get it wrong there. In 2011, the National Academy of Sciences, the United States National Academy of Science, an august body, meets to plan NASA missions for 10 years. They mention Viking in one paragraph, and it's like reading an essay of a high school kid who went in to take the test without studying. Okay. They are aware that Navarro Gonzalez has interpreted the Viking data in light of perchlorate. They cite him, but then they get it exactly wrong. What they say is exactly the opposite of what Navarro Gonzalez says. And that is what guides the 2013 to 2022 decadal survey. So for 10 years, NASA is making mission decisions based on a term paper from a student who hadn't studied. And it's very entertaining. It really gives you the insight into how high stakes science works when it's done by real human beings. I mean, none of us are very smart compared to the problem that we're being asked to solve. And so, yeah, and also when you start getting communities of science and getting committees of sciences, the ignorance amplifies, right? It gets reinforcing. So what you're looking at is documents that came out of NASA. Well, actually, they're not out of NASA. They're out of the National Academy of Sciences. NASA merely commissions the study. They don't actually guide it. So this is the community basically saying that they don't understand the first thing about chemistry that is necessary to do the mission, which we now call astrobiology. So it's quite surprising. But if you put yourself in the situation, you might have done the same thing, because you would also be afraid to say, "Hey, there's life." But yeah, you're right. Why go if you're going to find life, but you're afraid to say it? Why go in the first place?
Don't spend the money, and then once you do it, and you get a result that you've talked yourself into being ambiguous.
Yeah.
Send another one. Send a better one. Look for things like chirality, things like that, other things that you can do. Don't follow the water, which is exactly what they did. They dumbed down those experiments afterwards and just looked for evidence of water, even though the surface of Mars is covered with obvious evidence of past liquid water.
Yeah, well, even near surface ice is also abundant at this point. Look, I mean, you have to sort of understand. I've talked to many people at NASA. Now, remember, NASA is building a logo, an administrator. Almost all of what it does by kicking these things out to the community and out to the National Academy of Sciences for direction. And this is a problem, because if the National Academy of Sciences, the people on the Space Studies Board, which writes the decadal surveys, are not conversant with the chemistry that is necessary to understand their mission. I mean, it's like they say, the Pope will not be a Catholic if the cardinals are all heathens, right? You're not gonna get out of the National Academy of Sciences and the decadal survey sensible guidance for NASA. But NASA follows that. So if you wanted to write a grant application yourself to NASA, if you do not cite a chapter, verse, and even a line number of the decadal survey saying that you are doing what the 10-year plan says you're supposed to be doing, you won't get funded. They won't even bother to review your application. We're very fortunate because there's something called the NASA Innovative Advanced Concepts Program. It's got a separate budget. It has a consultant, a guy named Ron Turner, who I love to talk to, because he tells us that the NIAC, NASA Innovative Advanced Concept Program, is designed to break the decadal survey. Right? They're supposed to be putting money into areas where we're not endorsed by the community. And so they've actually supported us. They've actually supported here in my lab, a man named Jan Spacek, who is building what is called an agnostic life finder, or ALF machine. And so there may very well be a mission to detect or go seek life on Mars where you need a ride. But maybe Elon Musk or the Chinese National Space Agency or somebody will provide us one. But the bottom line out of all this is that you do go because over time you hope that sensible people come along and direct resources. I mean, it's just interesting. Just a couple of days ago, the NASA, the administrator, Jared Isaacman, just endorsed a proposal from the Italian Space Agency with Schmidt having provided a rocket launch to send 3 robotic missions to Mars. And they are the usual ones, measuring the atmosphere properties and these types of things. But because nobody understands that there's a more likely than not that life exists there, because there were no organics in the soil and the soil is self-sterilizing, even though we've known for more than 15 years that that's just not true. But because the culture and the community and the advice that goes to these national space agencies is not going to move, right? This is Max Planck, right? Science advances one funeral at a time, right? You're not going to get but this is the whole point. Keep in mind that, I mean, this is not unusual in science. I think that's one of the things that your listeners really should understand. There was a time when, you thought that ulcers were caused by stress and spicy food. All right. Now, that was not true, but there were billion-dollar molecules sold as drugs to cure ulcers by reducing your spicy food or your stomach acid. Now, we know now that ulcers are caused by bacterial infections, at least most of them, in your stomach. And that was done only because, a couple guys had the idea. They couldn't get funded. They self-experimented. They eventually did win a Nobel Prize for that. But the bottom line is that it happens all the time. Now, the only difference is that if you wanted to study ulcers, you could this afternoon go out and find somebody who's getting one and study it, right? If you want to study life on Mars, historically, you couldn't get access to the subject matter without having a national space mission supporting you with a consensus behind it. Well, science is becoming more democratized. There is actually the guy I mentioned a moment ago, Jan Spacek. He has got an international Mars penetrator rideshare system, where the idea is if we can get Elon Musk to agree to send a bunch of these to Mars, 1,000 of them, you drop them off, you don't have a soft landing, you have aero braking, but you hit the surface of Mars at 20,000 or 30,000 gravities. So you penetrate the surface. You have to harden your equipment so that it survives that. But penetrators have been used on many occasions. But now you can have something going to Mars for not $25 million, but like $25,000. And your Cub Scout troop could raise that amount of money in principle and design one of these Impress planetary penetrators to do an experiment with life detection, mineral resources, in anticipation of human arrival, something. It's up to you, but the only requirement would be that it be sterilized so you don't bring life to Mars that you don't want to bring. But otherwise, this is the democratization of Mars exploration. But again, it requires a ride share, right? So you have to amortize an $80 million launch or $100 million launch, whatever SpaceX will sell it to you for, over 1,000 or 10,000 different experiments, each being done by a Cub Scout troop or a Girl Scout troop or maybe a college class or a high school science class. And so if you amortize the launch over 10,000 probes, then the individual experiments become much more accessible. And that's democratization. It doesn't require consensus. It breaks a decadal survey. And it's done by people, including your audience, who know more about Mars than members of the august United States National Academy of Sciences, which is quite a statement.
Yes, but it's very true, and we do live in an age of democratization. Everything, from the media to, whatever, for better or worse. But at the same time, you're right. Just about anybody can pay, to have an experiment tag along on a large rocket, and you can send up CubeSats or whatever. Yep. And, you can go to Mars at this point, and you can do your own Life Finder experiment. But if you have 100 different Cub Scout troops sending Life Finder experiments to Mars and they all come back positive, at what point does it overwhelm everything? And one must say there is a preponderance of evidence for microbial life on the surface of Mars.
Yeah, no, but you're being very intelligent here. Keep in mind that, I had a fun a couple of weeks ago in Madison, Wisconsin. The Astrobiology Science Conference got together, and Becky McCauley-Wrench, who is the head of the astrobiology program, was on stage. So I asked her, has she told Jared Isaacman that life, more likely than not, was discovered by Viking? And she said, no, she hadn't. Okay. Because she had seen no convincing evidence or no convincing proof. So then, I had a private conversation. You used the word preponderance of evidence. That's the court case, right? I mean, beyond a reasonable doubt will get you sent to jail, but, by a preponderance of evidence is how you decide to go back and look again. And you're sitting here in a situation where for 50 years, except for the fact that they misinterpreted one lousy GC mass spec result, primarily because they did not apply intellectual rigor to the life negative interpretations at the same level that they applied to the life positive interpretations, and therefore screwed things up. For 50 years, what you have set with a preponderance of evidence that Viking found life on Mars, which doesn't mean that you write it into the textbook. It doesn't mean necessarily that you give it a species name, but it does mean that you go back again and look better. And as you said, when I was in Europe, I came back to the United States. I was an inaugural member of the NASA Astrobiology Community Institute, actually, in 1999, and remained so until it closed 20 years later. But the first thing that they told us that you were not going to go look for extant life. You could follow the water, okay? You could look for habitability, which in itself is a bit of a weasel word, right? It's the idea that life could be there or could live there without actually saying whether life is there, which is, of course, an interesting way of going after it. But, it's interesting. In 2022, 2021, 2022, I think the report was released in 2022. Vicki Meadows, who's an astronomer, and Heather Graham, who's a geologist at NASA Goddard, they ran a committee to come up with a list of what everybody would agree you were. Well, what they wrote was that they would come up with community standards to minimize the impact of a life detection claim. I think I'm quoting them verbatim. So that is what they wanted to do, was set up standards so that if you said, I have found life on Mars, we could minimize the impact of that statement on the community, which is a curious way to approach science, right? I mean, I can think of standards to try to decide whether or not your analysis of data are better teaching students statistics. There's all sorts of things I can imagine. But they wrote a whole section explaining why they thought that these standards were necessary. One was a discussion of Viking, and they get it wrong. Again, they say there's a strong oxygen in the surface and so on. And so the bottom line out of all this is, well, this is a Feynman line, right? Richard Feynman, the physicist at Caltech. Richard Feynman once said that science begins with a distrust of experts. Okay. Well, here you got it. It's in spades. But look, I mean, the democratization of this is very important. It's very important. So we have gotten this book, which you have had a look through. It's just, it will be appearing in about 4 weeks. I just got the copies today from the United Kingdom. It's being released by Penguin Press. And it goes through the whole story of what went wrong with Viking, with a sort of a sense of humor, because it's the only way you can approach this. Science is a human activity where we humans are ape descendants. We're kind of funny, especially when we interact with this high stakes science, because the culture has a back and forth that you would not necessarily expect if all you knew about science was what you learned in your 8th grade science fair.
Yes, Meet the Neighbors, is that the title it's going
That's
to?
Right. Meet the Neighbors, Life on Mars and How to Find It.
Life on Mars and How to Find It. You needed to write this book in 1965.
A little too young.
Yeah, well, negative numbers for me. So I knew.
Well, I didn't say that. I was said that. I mean, I also have written a couple of peer-reviewed articles on this, and Chris McKay, who's a buddy of mine at NASA Ames, who's a very, very bright guy, and he's taught me almost everything I know about the desert in Chile. So he said, "Why hadn't you written this before?" And the answer that I give is I did, right? So in 2000, I wrote it. In 2007, I wrote a whole book on the subject called Life, the Universe, and the Scientific Method, where I have a discussion of the Viking results. This is before perchlorate, so I was also not smart enough to think of perchlorate. And then again, I've written blog posts and peer-reviewed papers, and right. So what's different now is that we've decided that it's time to take no prisoners, right? So we are talking to you. We'll talk to anybody who will talk to us. We're running. There are pieces that are coming out in Science Magazine. We put a piece in basically getting them to agree to sort of retract or at least correct the mistaken statement from Klaus Biemann from 1976 that methyl chloride is a terrestrial contaminant. We have Scientific American is doing a piece. There will be conferences. One is being arranged in Williamsburg, Virginia, at William and Mary. So right now it's a full court press. We are writing editorials. I've got Ron Levin, who's Gil Levin's son. Gil Levin was again the guy who ran the label release experiment looking for respiration on Mars. And I've got a bunch of science bloggers who understand this. And it's very important, I think, to teach the science, because what you don't want to do is have the story be Stephen Benner says that, or Gil Levin says that, right? I'm an educator. I'm not going to be happy leaving the room if I haven't made sure that you understand all the details of the chemistry that go into the analysis here. Because the chemistry is, well, people think chemistry is hard. It's not that hard, but it's important that everybody understand that this is not somebody's opinion. This is the result of objective and logical analysis of what was observed.
Now, I had the privilege of interviewing Dr. Patricia Ann Stratt before her death. Just probably the last interview, months. And she was the principal investigator from Johns Hopkins and on the labeled release experiment, along with Gil Levin. And she was convinced that, to the very end, that they had found evidence of life. And I think that what bothers me most about the labeled release experiment, especially, but all of them at this point, is that it's evidence. Aren't you supposed to be evidence-based scientists? That there's just a gun-shy, people get scared when you start talking about aliens, and especially an alien microbe on Mars. It's like, yet it's certainly not impossible that there was a second abiogenesis or one abiogenesis and it was deposited here and it became us, or it's panspermic life from Earth that ended up there at some, descendant of E. coli or something that meteorite blasted off a dinosaur or something like that and landed and,
there certainly has been exchange back and forth. In fact, I have 5 grams of Mars from Black Beauty, one of our donors, a guy named Robert Bruner. He has since passed away, but I run a nonprofit foundation for applied molecular evolution here in Gainesville, and we have various people donate things. Money, of course, is always appreciated, but he donated 5 grams of a meteorite called Black Beauty, which is from Mars. It's what they call a breccia, one of these conglomerates, a bunch of pieces of rock together. So there has been traffic back and forth. And so there are bits of the meteorite that, the goo that was ejected into space, not a lot of it, from the meteorite that allegedly killed off the dinosaurs on Mars. And so there's traffic back and forth. But yeah, I never had the pleasure of meeting Patricia. I do have her book. She wrote a very nice book called To Mars with Love. And it's interesting the way you phrase it, because I think that's how she phrases it. There is evidence for life on Mars, and full stop. Now, I can go and explain to you why the label release experiment cannot be correctly interpreted as she would like to interpret it. There are a couple of small problems with this experiment, one of which is that they put 7 examples of food onto the surface. And without going through them, they have glycine, which is an amino acid. They have alanine, which is an amino acid, which you've already mentioned, chirality. It has left and right-handed forms. All of these molecules require that you break a carbon-carbon bond to release carbon dioxide. One of them, which is formic acid, which is 2 hydrogens, 1 carbon, 2 oxygens, does not need to break a carbon-carbon bond to release carbon dioxide. You only have to break a carbon-hydrogen bond, and that's easier. And so if I had designed that experiment, I would have left formic acid out and just gone with the amino acids. They also have lactic acid, which is what builds up in your muscles when you exercise without too much. So if you're not fit in particular, that also comes in left and right hand forms. So it is conceivable. I can't tell you the exact chemistry that does it at the rate that the Levin-Strauss experiment saw, but it's conceivable that if I had to find a reason why that would be a life negative result, that's where I would go. The same thing for the photosynthesis. The problem with the photosynthesis experiment primarily was, well, they didn't have a chemist on the design. That's a paid political advertisement. But they did not expect perchlorate. They did expect nitrate. They actually saw 100 times more organic carbon fixed by photosynthesis than they thought they had. And that's because they saw a huge burst of carbon dioxide coming off the surface when they heated their sample to try to get the organic material off. I would have added an acid wash step up front to try to make sure that carbon dioxide is not absorbed as a carbonate, like sodium carbonate in your toothpaste. So there are details. We can go back through each of these experiments in some detail. It's better to do this in written form with a blackboard. It doesn't work good on a podcast. But the bottom line out of all this is that, yeah, there is evidence that there was life on Mars. There are little bits and pieces which we would have done differently, one of which you've already mentioned. That is, we could have sent the left-handed and the right-handed food separately because your body only handles one of those two. It doesn't handle the other. And almost all life forms, even if they can handle both of them, for which there are very few, they handle it at different rates. And so non-biological oxidation, generation of carbon dioxide from that soil, if it's the same for left-handed and right-handed food, it would be more likely non-biological. If we're different for left and right-handed food, it would be most certainly biological. And that's the kind of experiment that actually, it's interesting, Gill did design that experiment himself. He actually flew that experiment. The Russians persuaded to launch that experiment, which unfortunately the launch failed, and the rocket and the experiment came down in the Pacific Ocean, a bit of Bolivia, and I think some of it actually also hit the Atacama Desert in Chile. So that's a bit ironic. But the bottom line is the mission failed, but they could have done that experiment any of a number of times on any of the rovers, the Perseverance, the Opportunity, the Spirit, Curiosity. All those experiments, we could have done life detection, extant life detection, improved based on what we know over what had been looked before. But the decadal survey is written by the National Academy of Sciences, the Space Studies Board, and the Space Studies Board in 2013 was clearly not conversant with the science that they needed to be conversant with to make sensible decisions.
Which is rather strange because in any other area, scientists like to chase Nobel Prizes. Not this time. There is some kind of a deep-seated bias regarding even microbial life on Mars, even though Mars just hints and hints and hints that something happened there. Something was active there at least at one time, if not now. And now we, in hindsight, we have all of these weird pieces of evidence that point towards the, basically fossil life. What is your opinion of finding what look to be microfossils, is the best I could call them, in the Allen Hills 84001 meteorite, which was the second, we haven't even gotten to this one yet, the second result that seemed to indicate that there had been life on Mars based on meteorite evidence.
Right. So for your audience, Allen Hills is a place in Antarctica where they sent a mission down to collect meteorites, because if you see a rock sitting on top of an ice sheet, it fell there. If you go see a rock out in my backyard, I'd have a hard time knowing that. The same reason why so many of these meteorites that you can get off of eBay are coming from Northwest Africa, because they fall into the Sahara Desert. So they picked up, and it's Allen Hills 84001, so it was picked up in 1984, and then a guy named David McKay at Johnson Space Center in Houston broke it open and saw structures which were then published in 1996 in Science Magazine, which looked as if they were cell-like structures. They were carbonates, they were fossils, of life. They contained organic carbon. At least Dick Zare and his group at Stanford got mass spectrometry. There is mass spec again off of these, and so there was also in that rock some magnetite, which is iron oxide Fe3O4, which is not rust, but it's a little bit less oxidized than rust, and so it's magnetic, as the name magnetite implies. So they were also found in that sample, and some people, Joe Kirschvink at Caltech in particular, wanted to interpret those as the results of magnetotactic bacteria. So we have on Earth bacteria that make little crystals of magnetite in them, and they use that to orient themselves in the magnetic field, that is Earth's magnetic field. So the Allen Hills meteorite had those as well. So what happened was that they discovered later that the organic molecules were not actually Martian. They actually had the carbon-12, carbon-13 isotope ratios of Earth material. So it just goes to show how much these organic molecules, which are basically derived from the burning of coal, so they're soot, make it all around the planet and end up in this. The magnetite crystals were a problem because the argument had to be that they have to be a certain size and shape, because natural magnetite that's not biological is frequent. The biological magnetite that we see on Earth is all of a very defined size, so that it's super paramagnetic is what we say, and it's of a certain shape. And what happened was that you had to cherry pick. You had to look through the entire assembly of all the magnetite crystals to find 3 or 4 which you would then show as the ones that look biological in an ocean of molecules of magnetite crystals that were not biological because they didn't have the right size and shape. And so at the end of the day, that story fell apart. The cells were a problem because if you looked at them this way, they looked like cells. They were small. Much smaller than the typical bacteria cell. So I wrote a paper at one point trying to argue or analyze how small a cell might be in order to support life, and came up with a constraint. We had a metabolism of 50 different steps that could go into something that small, but otherwise it was hard to do. But then if you look at the same structures from a different direction, they looked a lot more like crystals of minerals that would grow. And so, I'm a chemist. I've already told you my bias. I am very much hesitant to look at shapes of things and use them as a biosignature, especially when I don't control the survey. Because if I look at with an electron microscope at a piece of plastic, sooner or later I will see things that remind me of Mona Lisa, right? And so it's very much of a problem where the eye will see what we want to see. I mean, the classic example of this for Mars, of course, is the observation of canals on Mars, right? We now know that there are no canals on Mars, that basically there are dots, and the human brain will put lines through dots if you look at them at the right resolution. So, I think at the end of the day, the Allen Hills meteorite is a demonstration of the possibility of panspermia, that is, interplanetary panspermia. Avi Loeb, my buddy at Harvard, thinks, of course, of interstellar panspermia, but we certainly can have local panspermia. So it's quite possible that life arose on Mars and came to Earth, or life arose on Earth and went to Mars at a time when Mars was habitable, and more than it is now, at least from a human perspective. So I'm not a big fan of Allen Hills any longer. We did our best, but I had to change my mind.
Now, there are those on this world, Elon Musk. I don't want to name names, but we can say Elon Musk, that want to go and build the canals on Mars and have humans go there and start mucking around in the Martian regolith. And the bottom line is that a bacteria, which is what we're probably looking at here, not a virus, bacteria. And bacteria are very strange things because they can be very, very persistent. It's called an infection. And is it possible we could dump somebody on the surface of Mars and they get sick from our lack of not, recognizing life when we should have?
Yeah, look, I mean, the part of me that is not a chemist is a biologist, and we do know that parasites and viruses are an excellent example of this. Track with the organism. So a virus will evolve with an organism and its host. It co-evolves with the host. And so I'm not really particularly worried about Martian viruses infecting humans. As I say, we have many grams of Mars sitting in my office right now. So there certainly is the opportunity for bacteria from Mars to come to Earth. But in general, as long as, if they don't share much of a natural history, right, it's very challenging for a Martian organism to survive on Earth food and vice versa in environments. Now, that's so I we know Robert Zubrin and I wrote a paper on this subject. It was published a couple of years ago. Robert is the head of the Mars Society, and so it's really not easy to have. So you won't be infected by bacteria that even infect relative. I mean, you certainly won't be. It's very unusual for you to be infected by a bacteria that is infecting something quite distant from you. And so I'd be very much surprised if Martian bacteria caused human disease. Of course, it will not be the first time that I'm surprised. So it is, of course, absolutely essential in my view that we go as soon as we can, 2028, if we can get the funding. As you know, the most important thing in science is money. So no bucks, no buck, no Buck Rogers, I think is what Tom Wolfe said in that book, The Right Stuff. But yeah, it's very important that we go before humans go to look and understand as best we can the astrobiological potential of the Martian surface. But yeah, that requires, of course, again, the democratization of science will be part of this. If it's going to be a NASA mission right now, the Decadal Survey was written in, now this is not the 2013-2022 Decadal Survey. This is the 2023-2032 Decadal Survey. They do have a mission to Mars to look for life, extant life. But if you actually look at their projections and their budget, it's 2039, 2040, 2041, right? Way into the future. And so, if the Chinese National Space Agency does, even if Elon Musk does what he's planning to do, NASA's search for life on Mars, according to the current Decadal Survey, will be irrelevant. But hey, this is the point. You do want to know before you go, right, what the astronauts are going to encounter, because the last thing you want to do is have the astronaut get sick on Mars. You would not know a priori whether or not he got sick because he had a bad genetic mutation, because whether one of the bacteria that he carried with him from Earth. I mean, remember, Matt Damon grew potatoes in his feces, right? Or you won't know if he has gotten something from Mars. And so you're in this situation where without a context, you would not know whether that astronaut would be allowed to come back.
Well, that's the question, isn't it? Because there are unknowns everywhere with this. And I worry, the reason I asked the question was I worry about a dying planet. Mars has been through a process of death, effectively, the water going away and all of the things that happen there, the drying out. And it seems like it just, things could generalize, biology can generalize in a situation like that. We've never seen that before because Earth hasn't been through it. Even at the worst point, one day after the dinosaurs die, it didn't get that bad. No planet, this planet has never been so bad, for life as Mars is. And although I don't want to say that too much because it could have shallow, beautiful, gorgeous aquifers running through it for all we know. That could be really shallow. And a lot of people don't realize this. You brought this up in the book that, there are places on Mars that can still have liquid water under the right conditions, under the right pressure and temperatures. And Mars gets, outright balmy at the equator. I mean, it can get up to 68 degrees Fahrenheit. And, I mean, this is not as totally harsh as you might think. Go into that.
Yeah, well, let me change your perspective a little bit. I mean, keep in mind that the Earth started out with an atmosphere much like Mars, mostly carbon dioxide, some nitrogen, but no oxygen, right? Oxygen is only emerges about 2.5 billion years ago as a result of large-scale photosynthesis. Now, oxygen is a poison. In fact, the only reason why you think oxygen is good for you is because you have gotten mechanisms to manage the toxicity of oxygen radicals, which you can read about in your health food magazine, right? I mean, the oxygen. And of course, you still have anaerobic organisms. There's a large number of organisms that will retreat below your gum line to cause gingivitis to get themselves out of the oxygen, which is poisonous in the air. So you can sort of imagine the condominium association in 2.5 billion years BC complaining about these cyanobacteria who are polluting the atmosphere with this poisonous oxygen. So that's been a dramatic change in the Earth's environment, which now, I don't know if you're a big fan of Jurassic Park or not, but there is this line that Jeff Goldblum has, which is, I think, if I quote, life finds a way, right? So what life on Earth found a way to manage the oxygen, which is poisoning the atmosphere, which is a dramatic change. Now, admittedly, it's a surface change. If you go down a couple of meters, and certainly a couple of kilometers, you've got no oxygen that you need to worry about. Same with Mars. I mean, there's no question that Mars has lost its atmospheric pressure and it's lost its liquid water. And even in the balmy 68 degrees, because the pressure is so low, even if you transiently get water, it will evaporate quite quickly. And so, yeah, did life find a way, as Jeff would say, to adapt to that? And it really depends on your perspective. We do know that on Earth, every time we go to an environment which we consider extreme, like the top of an Antarctic mountain or the Chilean desert, we think that's an extreme. We would not be able to survive without the accoutrements of civilization to support us. But, life has found a way to survive there. If it's got energy and water, it's survived. And so the question you have to ask is the same about Mars. Now, again, go down below the surface. No question about it. Life could have retreated to below the surface and survived there, just like the bacteria do in your gum line. So it's a interesting question. Now, keep in mind that there are regions on Mars, which we call special regions. There are regions where we think that life from Earth could survive. Therefore, we don't go there to look for life because we're afraid of contaminating it. But there are planet-wide dust storms that cover the entire planet with dust, and these have got to be moving material from the special regions to places where NASA puts down a Viking lander, which is not, by the way, in any sense, a place on Mars which is high on my list of habitable spots. But yeah, they put. So the point is that life, if it's on the surface in any of the habitable regions, would have been blown in there. And so the question is, it's going to have a reason to want to be dormant. It probably would be dormant. That's what it is most of the time in the deserts of Chile or Antarctica. But, Viking comes along, it heats it up to a balmy 10, 15, 20 degrees centigrade. Not very hot. They put it under pressure. They give it water. They give it food. And now, my only problem, if I were that life form, was that if Viking dumped, I'm sitting in my sunroom doing the crossword, and Viking then dumps a whole bunch of food on top of me, I might very well find that food poisonous, or the water drowning me, or something like that. It's unusual. But yeah, I would not be surprised at all if life exists in the special regions, gets distributed, there's a good survival reason for that life to go dormant, and to be only coming out of dormancy whenever it has the opportunity to do something productive. We see that in all sorts of environments, extreme environments on Earth. This is one of the wonderful things that the NASA Astrobiology Institute did do for us. We have a much better understanding of extreme environments on Earth than we did 20 years ago. So yeah, I mean, I think it's possible.
Now, about this idea of a dying planet, things like food might get scarce and water might get
Yeah.
scarce, and life might go in the direction of living on less and less, which means that if you put too much in, you could kill it. Did the Life Finder, experiments on the Viking landers, could they have accidentally killed the life
Yeah.
just simply by putting in the wrong amounts of water?
I'm with you. No, that's another excellent question. Yeah, for your audience, the photosynthesis experiment was run by Jerry Hubbard, George Hobby, and Norm Horowitz, who I've already mentioned, and they had a team, of course. They argued that their experiment was the best. And the reason was because they did nothing unnatural to the soil. All they did was replace the carbon dioxide in the atmosphere. There's also a trace amount of carbon monoxide. They replace it with carbon-12 as the isotope by carbon-14, which was radioactive. Otherwise, the pressure was marginally higher, not out of the range of the pressure variation that you see on Mars. The carbon monoxide ratio was a little bit higher, but not all that much. And so they did nothing to that surface that might be regarded as killing anything. They really didn't. The environment was. Now, they did do two other things. They brought their own light bulb along. They were afraid that if they let the sun filter into the soil, the sun is not, there's no ozone in the Martian atmosphere, so there's nothing that screens out the very harsh ultraviolet light. And they were worried about that. So they actually gave the Martian organisms some very pleasant light in the correct wavelengths, not very hard ultraviolet. So that was actually something that they may have done to benefit Mars. Now, of course, if the Martian life has adapted to, harsh UV, depriving it of harsh UV may be a bad idea, but that's what they did. So the other thing that they did, of course, it was a little bit warmer than the ambient temperature on that surface. And so, so those two things, however, were it was not extreme. They eventually did heat the sample to 170 degrees to see if they could kill the result as a way of establishing that it was a biological result, not a non-biological result, which would not be expected to be killed by heat. And they killed it, by the way. So heating Martians to 170 degrees centigrade evidently kills them. That's 400 Fahrenheit for your imperial units listeners. So yeah, that's pretty hot. But the bottom line is so the argument would be for that experiment, the answer is no to you. We did not kill that. We didn't kill the Martian life. We are looking at photosynthesis under very, very normal for Mars conditions.
Can you intentionally kill the Martian life? So let's do our Life Finder experiment again, to go through all of the motions. And at the very end, let's inject the Earth's atmosphere, the oxygen, the 21% oxygen into the experiment and see if everything dies. And all of a sudden, does that nail home, or could it potentially nail home the idea of anaerobic bacteria on Mars?
John, I don't know that I've actually seen that suggested. That might be a suggestion worth pushing. Of course, people have taught about heat as a way of sterilizing the surface, but oxygen does sterilize things, right? It's not a good sterilant on a life form that has seen it. Mars does have a little bit of oxygen. I think it's 0.17% or something like that. It's of course, but there's a little bit. But if you were to boost oxygen to 20%, Earth levels 21%, something in that range, I don't. It's a good question. I haven't thought it through, and it's something that I don't think has ever been suggested. But you should suggest it. Write a paper.
I will contact the National Academy of Sciences and hear back.
Yeah. Well, Gil Levin talked about how, and I quoted his anecdote in the book about how he talked to the National Academy of Sciences about life on Mars, and he said they were about to throw shrimp from the shrimp bowl at me. They said that you have disgraced yourself, you have disgraced science. So expect that to be your reception.
Hey, I'm a science fiction author. I don't hold no PhDs, so they can throw whatever they want at me. I want to take the Earth's atmosphere to Mars, because that's exactly what we're going to have to do.
Yeah. Well, that's right. Well, I mean, in a sense, you could argue that is a protectant for the astronauts, right? The fact that the astronauts need 20%.
The fact that Earth exports its biosphere in the form of us and our oxygen sealed environments. If it's self-sterilizing on Mars, that would be useful to know.
Yeah, that would be very useful to know. That's a damn good question. We should. That is more than a good idea. I think that's an excellent idea. That's a very easy idea to implement. Okay, so you could, with much lower power than you needed to heat the sample to 170 degrees centigrade, you could, with much lower power, turn on a valve and put oxygen in to see if the result went away. I mean, this is the case. I mean, if you go to Cleopatra's tomb or someone, you pull a papyrus out after 3,000 years, some pharaoh, right? It's going to crumble. If it has been exposed to air. No, it's not fast. I mean, obviously, if you heat the papyrus up in air, you set it on fire like the perchlorate did for the GCM spec. But your butter goes rancid, your gasoline goes stale if you leave it open, the can open to the air. Oxygen will over time destroy even Earth life, and the reason why you're not destroyed, well, in fact, you are. Okay, you're not immediately destroyed because you have enzymes that manage the radicals and all the rest of it. But if I put you in an oxygen tent because you are, you have lost a lung capacity for some reason, if I give you super oxygen over time, it's bad for you.
Well, there was a case in the early 1980s where they had found a medieval body. It was St. Bees man, for those that like to look stuff up. And it was a gentleman that almost certainly was a nobleman named Anthony de Lucy that died on a crusade. And they sealed him in a lead coffin. They basically canned the guy and possibly even cooked him to make sure that he would preserve on his way back to England. And when they found him intact, and they dug him up and they opened up the lead can, and he was basically as preserved as one can be, having been canned in the year 1350. Okay. And they removed his liver, and they started sectioning it to examine it, and it was literally oxidizing before their eyes, going from an astounding pink color, like what might have been in life, or what was in life, to brown, and it was doing it in seconds.
Was the enzymes in the liver still active? Did they? I'm sure they.
I don't think they got that far, because I think they had taken the body to a local hospital. To do a cursory examination. I don't think they got past cursory, because the hospital was like, get that old dead body out of here, and they reburied it. But had they done that, it would have been an interesting result. But I never saw anything that said that.
I should look that up. If you ever get to the panhandle of Idaho, you get to a town called Clarkia, and on the outskirts of town there is a guy who runs a racetrack, where he had dug into a bank of 22 million year old clay, which had preserved water from 22 million years ago in the bottom of a pond that had leaves in it. They extracted the leaves, they opened they break open the clay, and the leaves are green. And then within minutes, they turn black.
It doesn't last, and oxygen is perfidious in this regard, and that's why I was thinking about that experiment. I was like, oxygen might be useful, and I would love to know, what an anaerobic, completely second abiogenesis from Earth life, unlike our anaerobes, practically, although probably very much like them. would do if it suddenly got oxygenated. We would be the great oxygenation event for Mars, especially if we terraformed it. We'd
Yeah, I hadn't thought about that. No, the Martian Condominium Association would certainly object to this.
Yeah, I don't even I don't care about the Martian Condominium Association.
You obviously don't own a condominium, and you've never had to deal with one of these associations. All right,
no, I have dealt with HOAs. Almost as bad. Now, what is the path forward? All right, tentatively biology on Mars. Lay out your ideas for future experiments that could, lay some of the problems with the Viking experiments to rest in a modern context. That say, all right, we can go there, we can do it robotically, we can test for life, and we can reasonably walk away and say, yeah, it's there. And
no, I'm with you.
Taking into account the naysayers that are, of course, going to show up and say, no, you didn't find life.
Yeah.
And it'll be round 2 at that point. But what's the future here?
Yeah, so I mean, as I mentioned, the most important thing in science is money. So I will structure my answer with increasing expense. So you got to keep in mind that I also do biotechnology and license diagnostics and biotechnological tools. So I have some resources. I'm not Elon Musk, but so we're putting in $50,000 to go back and do a whole series of experiments, which would add value to the Viking experiments, but without having to go back to Mars. So there are certain things that I mentioned to you, like formic acid being mixed in with left and right-handed amino acids. There are papers from NASA Ames, for example, where they have said that Gil Levin and Patricia Stratt incorrectly interpreted their results. We have to go back, and they think that perchlorate is responsible for them. That appears in official documents, including the Meadows-Graham document that I mentioned earlier. We would do life experiments on Earth to try to constrain those possibilities. That is, if you can think of a reason why the Viking experiments did not detect life, and that's a chemistry experiment, we go back into the laboratory and do that. And we're looking for funds to do that, as well as what I provide. The same thing for the photosynthesis experiment. I didn't go through a lot of the technical details, but there are technical details where if we did certain experiments on Earth today, knowing what we know about perchlorate and all the rest of it that they did not know in 1976, we can do experiments today on Earth without going back to Mars, which would increase the value of the data that we do have from 50 years ago, and to remove some of the objections to the life positive interpretation, or quite possibly amplify them, one or the other. So that's what I would do in the $50,000 to $100,000 range. Now you have to go to the next level. So the next level is what NIAC is supporting, or of course private donors have also suggested that they can support this as well, which is to try to build agnostic life finder instruments here on Earth to the point where they attract a ride. So you're not going to get a ride to Mars for $1 million, but you can build on Earth a machine that will look for the genetic molecules of Martians universally, because we've done a lot of what's called synthetic biology, where we have made alternative genetic molecules, alternative to the DNA and RNA that you and I have. And this is therefore defining quite broadly and quite universally what we think genetic molecules must look like on any planet in water. And so we have built a device with NIAC support to phase 1 to get this to the level where if we get phase 2 funds for this, we will be able to get it to the point where we can say, hey, we're ready to go to Mars. Someone who has the resources to launch, and the number of people there are not zero. There's a number of them. The Schmidt, obviously Jeff Bezos, Elon Musk, everybody wants to launch a rocket. So if we can get to the point where we attract that kind of a ride, that would be the next level. And Jan Spacek here is the guy I mentioned already a couple of times. He's the guy who's sort of developing that. Now, the next level, of course, is we have to attract somebody to do a ride share. Now, remember, the $3 billion, $4 billion flagship missions are expensive primarily because they have to do a soft landing. So this idea of using penetrators gets that mission down to not the 1 million or 10 million level, but somewhere into the 100 million level. And of course, if you do ride share, that cost can be dispersed, but you're now looking at something which is 5% the cost of a flagship mission, and it would drop penetrators onto the surface of Mars, where because you have a lot of them, right, you can afford a few of them to fail because they hit a really hard rock, or a few of them to fail because for some reason the mission fails. But you got 1,000 of them going, and so some of them are going to get through, and and so that would be the next stage. And all of that would get you to the point where we would be able, in a 2028 launch, given the funds, to be able to, by 2029 or 2030, when the penetrators arrive, to be able to say we have evaluated much more completely the astrobiological potential of the Martian surface. So again, we'd start with just in round numbers, $100,000, which is to, get the experiments done here on Earth to add richness and new interpretive value to the data that we already have from Mars. By the way, there's also data from Curiosity and the rovers on Mars and Perseverance, where if we had a little bit more Earth-based support, we would be able to add value to those from the perspective of biology. So that's 100,000. The $1 million range is, of course, trying to get one of these life detection instruments to the point where it attracts a ride. The $100 million would be a ride that involves penetrators, where that could be divided over 10 people, which is a $10 million, over 100 people, which is a $1 million per experiment, or over 1,000 people, which gets you down into the Cub Scout range. And that's what we would do next. And that all is before we would think of going to Jared Isaacman and saying, hey, you have to get a flagship mission for $5 billion to go.
Now, could you get a twofer out of this? In other words, say you have your penetrators and you deploy them and you're doing your experiments, but at the same time, you might be exposing subsurface liquid water, which on Mars is probably shallow, which would give you a profile of the availability of water.
Oh, absolutely.
You look for the evaporated, water vapor, and there you go. You get a profile while you do your life experiment.
Right. Well, the penetrators, I suspect that if I were to submit to 100 high school science classes a challenge to design a penetrator, I would get a lot of them coming in, which are prospecting the surface in anticipation of crewed landings that would be looking for resources. They would be water, of course, but you would also could imagine detecting copper or, any of a number of elements that you might find interesting, and prospecting the surface. One of the nice things about 1,000 penetrators is that you get 1,000 data points. With a rover, you put it down on one spot and then you have to walk around, and that's the billions and billions of dollars. So the penetrators would get you a lot of spots, and you would be prospecting for resources. You would be looking for water, like you suggest, or you could be looking for life.
All right, Dr. Benner, when does the Meet the Neighbors, the book, come out?
Well, Penguin Press is, there's a very nice man, Keith Mansfield, who has been the development editor for this book. It released on the 23rd of July, which is the 50th anniversary of Viking opening its doors for business on planet Mars. So there's a whole bunch of celebrations going on, of course, that are associated with this. I will be speaking at a number of them. But yeah, our goal right now is to say that 50 years of imbecility is enough. It's time to go get this show back on the road. And I cannot think of anything better than the 50th anniversary celebration to call public attention to this.
My last question for you today is this. All right. Do you think that outside of the box ideas in science are suppressed by a culture, and that, there is such a thing as stepping too far, even though you're within plausibility within science? And I think this was a case of that. was that people weren't, it's extraordinary claims, require extraordinary evidence was Carl Sagan's mantra, even though I don't know what extraordinary evidence is. I just know that you need evidence. And this was evidence, the Viking experiments, but it didn't stick. Do you think that there is a suppression there, a cultural suppression of so-called ultra ideas?
Yeah, no, well, it's well known. Okay, so two things. Of course, Carl Sagan, although I, of course, grew up with his television shows and his books, I think the statement of extraordinary claims require extraordinary evidence has done a lot of damage, because not only do I not know what extraordinary evidence is, I actually don't know what extraordinary claims are. And what you think is an extraordinary claim depends on who you are and what your background and culture is. So do you think it's an extraordinary claim that ulcers are caused by spicy foods? Well, I mean, it depends, right? So there are people who think how ridiculous it would be for you to assume that you did not have life on Mars, right? So this is a question of where burdens of proof lies. If nothing gets said, are you more likely to have Mars life or not? Well, the answer is that depends on who you are, right? And so, if you believe that Jeff Goldblum, that life finds a way, and you are aware of rocks going back and forth from Earth to Mars, there's life on Earth, you would be astonished if someone made a claim that life is absent on the surface of Mars. And that's exactly what happened, right? The claim that the Martian surface did not contain any organic molecules was an extraordinary claim. It was not recognized as such because, again, as I've already mentioned, organics fall to Mars all the time. It was an extraordinary claim that they didn't see them, but that's not how they saw it. So the problem fundamentally is that, yes, you don't know. Your culture determines what you think when you're not thinking at any level of depth. And so, and of course, the statement, I mean, there are books, Thomas Kuhn, The Structure of Scientific Revolutions. There are story after story. I have written a couple of history of science pieces. One is on the Krebs cycle, which you may have heard of in biochemical metabolism. I wrote a 20-page paper and published it a number of years ago, showing how the culture reacts to this and how people see what they wanted to see. You have this story which I told in the book, which you have seen, which is that Mercury has an odd orbit. We now understand that as the consequence of general relativity. Before 1915, they didn't know about general relativity, so they thought that there was a planet between Mercury and the Sun. They called it Vulcan, not of Spock, but that's what they called it, which was perturbing the orbit of Mercury. And what people don't know is that before 1915, there were many, many independent sightings of the planet Vulcan. Now, there is no planet Vulcan, but if you expect to see something, you see what you expect. And so, yes, look, there's no question that science has a long history of suppressing ideas that are out of the mainstream. Most of the time, the ideas that are out of the mainstream are wrong. That's why they suppress them, right? But you throw out the baby with the bathwater, as we say, right? The facts are that in order to get these ideas through, you need to have some degree of democratization of the science. When a single space agency driven by a single national academy with a single 10-year plan is doing it, that's the hardest environment to get an idea that is wrong, but is a consensus displaced by something that's right. And that's in a nutshell is what's gone wrong with the Viking results.
If everybody would like to donate to Dr. Benner's efforts to, a private life finder mission to Mars, where do they go, Dr. Benner?
Well, so this is a 501c3 organization called the Foundation for Applied Molecular Evolution, which has the initials FFAME, which is sort of like fame, but with an extra F. So if you send an email to info information, that is info@fafame.org, we can put you in touch with the person here. You'll get contacted back by somebody who is actually doing the science associated with the life-finding mission that we're operating here. So info@ffame.org is a great address.
All right, Dr. Benner. I must say that I can attest that as a person that really loves spicy food, especially a nice shrimp vindaloo, and I don't throw the shrimp at scientists, I have not gotten ulcers. I think it's actually probably the opposite. So thank you for joining us today. And
entirely my pleasure.
I look forward to many more years, Dr. Benner. Throw the wrenches in the works to get the questions flowing again.
Yes, and you can help.
I will.
And your audience can help.
Yes, if anybody